Not bad. Well, I think that we will get started. So, welcome to the ecological importance of fungi. This will be an awesome presentation by Bill Bakaitis, who is very much alive since he's going to be giving this talk tonight. So it's going to be a great lecture on the importance of fungi within the ecosystem. And so Bill's going to touch on a lot of different things. So first off, I want to thank you all for attending this webinar series on fungi. Some of you may have attended the one that was in May. If not, yeah, I believe I sent everyone a link to that YouTube video. This one will also be recorded and archived as well. So my name is Lauren Bohr, and I'm the Education Outreach Coordinator at Mohonk Preserve. And I'm going to be monitoring and facilitating this presentation tonight. So before I introduce our presenter, though, Bill Bakaitis, just a few reminders for everyone attending this. Please turn off your video and mute your audio. This is going to prevent distraction to our presenter and the others in the audience. So if you do have questions for Bill, use the chat box, which is at the bottom of your screen. You'll see a little chat box. I will actually put some links in that chat box to some articles that Bill has written. So the links to those, so you can check those out later. So there's going to be points throughout the presentation for questions as well to be answered, including at the end. So at the end of the session, we'll have a chance for any questions that may come up. So as I mentioned, this presentation will be recorded along with the chat conversation to share with our online presence. So please be mindful and respectful during this presentation and in the chat box. So video off and mute your audio as well. So now it is with great pleasure that I get to introduce you to our presenter, Bill Bakaitis. So Bill taught at Dutchess Community College for 38 years prior to his retirement in 2006. And during his teaching career, he was granted sabbaticals to study graduate level mycology, both at SUNY New Paltz and at the New York State Museum in Albany, where he worked with John Haynes, the New York State mycologist. He's a popular speaker. He's given educational programs in mycology at the Institute of Ecosystem Study in Millbrook and the Culinary Institute of America in Hyde Park, Hudsonia at Bard College, as well as many other institutions throughout the Northeast. In 1983, he founded the Mid-Hudson Mycological Association, which I think was even mentioned as we were chatting here. And since 1984, he's worked with the poison control networks throughout the Northeast. His articles have been published in the New York State Conservationist, Adirondack Life, Mid-Hudson Magazine, Poughkeepsie Journal, Mushroom, the Journal of Wild Mushrooming, where he is a contributing editor, and elsewhere. So now, without further ado, here is Bill Bakaitis presenting on the ecological importance of fungi.
Well, thank you. I'm going to share my screen with you all right now and hold control. This is the one I want. Okay, and I want to put it on. I've got to get this thing off the top. How do I get this thing off the top? I want to put it on. I got something on the top of my screen here. I can't get what I want to do.
Oh, good.
How do I do that? How do I want?
Oh,
no.
You are so close.
You can
at the top left-hand corner, do you see from the beginning?
Yeah, that's what I want to do.
Yes. So just click on that.
Oh, it does it without me doing the other thing. Okay. And
Yep.
how do I get rid of this thing on the top?
I don't think you do.
Okay. All right.
You
maybe
can see it now. It's fine. You're full screen.
Oh, really? Okay, I'm not here, but maybe that's because I was moving. Well, okay, well, here we are. It's so nice to meet some of my old friends. That's wonderful. Richard, I think you used to live in the Lower East Side, if I'm not mistaken, with I remember Wana Wu. Is that right? Oh, well, you can't talk to me now. Anyway, okay. So those of you who may have been around in the spring when we did the first of this series, will remember that this is was set up as a four or five-part series, introductory mycology for the curious naturalist, and I think a lot of you are that fits you to a T. You're very interested in what you see on the trails. So this is the second talk. It's ecological functions of fungi, and for those of you who did see the first talk, there were two important things that we want to just remember here and carry forward to this talk. You know, I am going to ask about how can I get rid of that black thing at the top? Well, maybe I can drag it. But what am I going to drag it to? It's still on the screen. Any way I can get rid of that? Anybody know?
So are you seeing
It says you are screen sharing, stop share, mute, stop video, participate, new share, pause share, annotate.
From the viewer, it looks totally fine. I think it's just on your screen, so you don't need to worry about it for us.
If I can, but I need to see what's down here when I point at something. Okay, let's see. This has never happened before.
Bill, you can try to hide the panel. So you have a view button.
View button. I don't know what it says. Mute, stop video, participants, new share.
View is on the upper right.
Upper right. Says more. Chat, record meeting, hide video panel?
View.
Hide floating meeting controls. That's probably it. Okay, that got rid of those. Okay, good. All right. So from the first talk, two really important concepts to carry forward here is to remember that fungi are different than you and I. They feed outside of their bodies, and that will be important when we come to talk about saprophytic fungi. And they have very distinctive sexual characteristics. And they don't have sex like you and I do, but they have, it's called a prolonged dikaryotic life cycle, mycelium. So you have your two spores from a mushroom. If they're compatible and are of different strains, different sexes, plus and a minus, they can meet as the spore germinates. They can meet and then fuse. That's called plasmogamy, and they continue on that throughout the entire life cycle. The juices meet. The chromosomes don't join. They float side by side through the hypha and mycelium, and even in the mushroom, until finally they get to the hymenium, which are the gills or pores, and that's where they quickly will complete the sex act. So it's a very long, prolonged sex act. And furthermore, the fact that two spores will join does not preclude a third, a fourth, or a millionth. So fungi are quite different with this very prolonged dikaryotic. There's the word right there, dikaryotic, two nuts, carry-on, two nuts. So it's N plus N. Okay, so those are two things really to hold in mind. We'll talk about those later. So this talk is going to be looking at the ecological functions of fungi. And here we have a typical northern Maine ecosystem, a lot of moss, coastal Maine, I should say. So a lot of things going on here. There are a lot of mycorrhizal associations, and mycorrhizae has become very important lately in a lot of the literature and a lot of the thinking about fungi. So we'll talk about the mycorrhizal relationships. They also have a saprophytic relationship. It's been said that fungi are the world's original recyclers. So that's an important kind of thing to talk about. But those are the two major things we'll be talking about. But they're also parasites. They are pathogenic. And then I want to introduce you to a new concept, if you don't know it, about endophytic. It turns out that fungi appear within the tissue of almost every plant on the planet. Or that's been said. It's probably hyperbole, but it just goes to illustrate that they live inside of plants all the time without doing much of anything. All right, so let's move on here. The first part of this is going to be a lot of talking, getting into some concepts, and then we'll have a look at some mushrooms as we get into this. So when we talk about an ecosystem, we're talking about not so much a thing, but a verb, not so much a structure, but a process. There certainly are structures, but more important are the ways that they interrelate. And it's often said of systems that within a system, everything is connected to everything else. So it's not connected in a linear way, but in a very convoluted way, lots of webs, and the properties there within the system are not static. They just change quite a bit. The other thing to say about this, just going along with that, is that these web systems relate one with another web system. So you might have an organism relates with other organisms, and that creates a larger community. And the communities will get together and they live in a biosphere. We live in a solar system, we live in a galaxy and so forth. And an organism within our organism, we'll talk about ourselves in a minute, but within our own bodies, we have lots of organs. Within the organs are lots of structures like an organelles, small structures that are there, chromosomes. Within those structures, there are hormones and all sorts of molecules. Within the molecules, there are atoms. Within the atoms, there are subatomic particles, electrons and neutrons and protons, all of which have charm and quark and whatever else are down there. So we're talking here about a lot of things that are interrelated. And the interesting thing about systems is that at any level, they have unique properties that are unknown until the system starts to coalesce. And then we say they have emergent properties. Now, all this sounds like it's chaotic, but it's not chaotic. It's rather regular in some way. And the way that it's regulated are by what we call feedback cycles. And these are self-regulation. It doesn't need someone from outside to regulate it. They regulate themselves. Homeostasis is a very good example of that. Let's talk about our own organism there in our body. Okay, so at the organismic level here at our body, let's talk about homeostasis. It's said that the average temperature of Americans is 98.6. It appears to be getting a little cooler. I know mine is now 97.4. Been regular that way for probably 5 or 10 years. Maybe it just happens when you get older. But within my body, I have a thermostat set at, let's say it's 98.6. That doesn't mean I'm at 98.6 all the time. And if you set a thermostat in your house at, say, 72, it doesn't mean it's at 72 all the time. If it's colder outside and the temperature is cold, then what happens is that there's an on switch that turns the furnace on and the temperature starts to rise and rise and rise and rise. It passes 72 and it gets up to maybe 74, 76, and then there's an off button and it turns the temperature off. It doesn't fall off real quickly, it gradually recedes over time. So over time, you have a periodic increase and decrease, increase and decrease, increase and decrease. And that's true of all of these cycles. What we call these are negative feedback cycles. They're self-correcting. They always have a tendency to return to the average mean. And if you were to mathematically, to say if you rotate that data orthogonally, and look at it, not as we're looking at it here on the sheet, but look at it from over here and look at it this way, then the data points will line up like this. Most of them will be right in the middle, and there'll be a bell-shaped curve more or less on either side. So that's where we get a typical bell-shaped curve. And if you're into mathematics or statistics, the central limit theorem applies here about how these make sense. So what are some of you think about what some of the switches might be in our body? Well, and we're talking here about the system that is our body. Well, one of the most obvious is that I have a brain, and my brain says I'm getting cold, and I can go and turn up the thermostat. I can put on a jacket. I can turn up the heat. I can get up and do a dance. I can do a lot of things. That requires my brain, and not all the brain, but specifically the non-myelinated tissue in the brain, the gray matter. And those fire by sodium potassium charge of about 40 millivolts, which run down the axon till it gets to the end where suddenly that electric charge is converted into a chemical charge, and vesicles release neurotransmitters that then turn on the next downstream neuron. And in my body, if I'm moving, all of those will turn on neurons that activate striated muscles. Okay, those are under conscious control, but there are a lot of muscles that are not under conscious control. They're under involuntary control, and those are controlled by smooth muscles. And there are a different set of neurotransmitters which activate smooth muscles. And smooth muscles will squeeze glands, and out of the glands will come certain hormones, chemicals, which will regulate the temperature, or the energy cycling in the body, and heat is generated. Glycogen is converted from the liver storage and is released into the bloodstream. There are hormones and compounds in the bloodstream, which then are connected to the brain in various ways. So there are many. We could spend hours talking about this. There are many ways that homeostasis takes place. Some are under conscious control, some are not. But most importantly, when we're thinking about things that don't have a conscious mind, we're talking about those involuntary control mechanisms. So, all in all, what we have here is a relative stability through what we might call redundant complexity. There's not just one switch, but there's a lot of switches. And a good mark of a healthy ecosystem is redundancy. The more redundant an ecosystem is, the healthier it is. A rabbit doesn't want just to have one kind of plant to feed on, nor does a deer, nor does something that would eat rabbits or deer. But the more diversified an ecosystem is, the healthier it is. So that's a general sort of take on systems concepts. Now, that cycle that we just looked at, that cycle that was in the body, homeostasis, looks very similar to the cycle here. There's another negative feedback cycle. This is called the Keeling Curve. Don't know how familiar you may be with this, but in the late 1950s, Charles Keeling wanted to get a measure of how much carbon dioxide was in the atmosphere. He knew he didn't want to go near cities, so he went out in the middle of the Pacific Ocean. Good, because there's a lot of algae there, which is going to recycle carbon dioxide as well. He knows carbon dioxide is heavier than air, so he wants to go to the top of the highest mountain. And on Mauna Loa, he sets up an observatory where he can measure carbon dioxide and content in the atmosphere. And within a year, he finds this cycle, this annual yearly cycle. And within a year, he immediately knew what was going on here. And here's the annual cycle here. Carbon dioxide in the atmosphere increases in January, February, March. Around April, it starts to flatten off. In May, it reaches its peak. In June, it starts to fall. July, August, September, October is about the lowest it is. And then it starts to rise again. And as soon as he saw this, he knew exactly what was regulating carbon dioxide in the atmosphere. Since I have this chart up, I'll just point out that in 2015, we were at about 400 parts per million. Last May, we were 414.7 parts per million. I've heard reports that, you probably did too, that last year was the hottest on record. So I'm assuming the CO2 was higher even than that. So if we were together in a room talking about this, I'd play with it a bit, but I want to move on because we have lots to talk about. So this is the annual cycle. And if you just look at this map of the Earth, this is a globe split open and spread on what we call a Mercator projection. So it's square. So there's the tropics are, the Arctic areas are greatly exaggerated. But even with that, here's the equator, and you see that the land south of the equator diminishes. the land north of the equator increases. So the CO2, if I just go back to this curve here, it doesn't matter where in the world you go, but you find the same signature. It doesn't matter where in the world you go, you find the exact same signature. And that tells us that the most important part of the globe, the Earth, the ecosystem for regulating CO2 in the atmosphere, natural, is not the equator, not subtropics, although they're important, but it's the northern hemisphere. And here we have a huge landmass, lots of grasslands. There's temporal and boreal forests there. And this gets at the importance of land-based fungi. So we'll talk about those now. Mycorrhizal, essentially mycorrhizal fungi, help the plants grow, and so they remove the CO2 from the atmosphere. But the saprophytic fungi, because they degrade the carbonaceous material, they add CO2 to the atmosphere. So that's the importance of fungi overall, and we'll now look at that in more detail. I rearranged these functions here just a bit, just to make a story. We're going to start with parasitic fungi, and we'll see that there's root rot that might topple a tree. And then once the tree's on the ground, you've got saprophytic action, and there are two major kinds of saprophytic actions that we're going to be interested in: white rot and brown rot. And they have different properties, and they're different fungi involved with them. There's also a soft or wet rot, and usually that's filamentous sort of slime fungi that down in the mud. The mycorrhizal fungi, ectomycorrhizal and endomycorrhizal are the two big ones that we'll talk about. There's also a number of others, but largely they're just variations of endomycorrhizal fungi. So we'll talk about that, wrap that up. I'll introduce endophytic fungi as we talk about mycorrhizal. And then finally, we'll end up by talking about pathogens of plants and animals, including fungi which grow in the lungs of people or in their mouth, and things you can see, mushrooms actually. So here we got the parasitic saprophytic process. It said that trees, because they renew their living tissue every year, are potentially immortal, not immoral, but immortal. So they grow year after year after year. But you might have something that rots the root, and that would be a pathogen that weakens the tree. Wind blows and the tree falls down. Well, it lies in contact with the ground, moisture gets in, and decay starts. And within a year, the cambium layer rots. That's the layer right under the bark, the vascular system, which carries a lot of the sugars. Within 2 to 4 years, the sapwood right under that rots. But the heartwood, the wood inside, often is of a different color, it is slow to decay, and it will take years, sometimes 100 years or more to decay. But when it does decay, when this does decay, there's two things that happen. To the extent that both cellulose and hemicellulose and lignin, to the extent that they are rotted, what's left is just little white shreds in the ground, and all of the carbon that's stored in the tree gets released as carbon dioxide. It's an oxidation process. The same amount gets released if fungi degrade it, or if you burn it in a stove. So this is combustion, slow combustion, that recycles all the CO2. There's another kind of fungus that will digest the cellulose and hemicellulose, but it doesn't digest the lignin. And the lignin is very complex molecule, multi-branched with arms coming off at odd angles all the time, and it's hard to break down. But so it stays in the soil, here not broken down, and it's a brown color, so it's called brown rot. And you see this in trees, spongy. You can pick it up off and squeeze water out of it, moist and nutrient-rich humus. And that involves a complex community of fauna, and it can feed the ecosystem for thousands of years. It said that about 2/3 of what we call soil is humus, and that's where the humus comes from. Humus in the forest or humus in the garden. in terms of applying these processes in other ecological considerations, you might use white rot fungi in what's called mycoremediation. You might use these fungi to get rid of compounds that are very complex, often created with high temperatures and just don't break down in the environment. Fungi which can break down lignin can break down these. So white rot fungi are often used in mycoremediation. As far as sequestration of carbon, storing it someplace safely so it doesn't get into the environment and cause global warming, brown rot fungi are very good for that because they put it back in the humus. Not only the tree's living stores carbon, but when it falls, brown rot fungi will hold most of it in the soil for a long time. So we talk about sequestration there and also mycoremediation. So, shall I stop here for a second, see if you got any questions, and then we'll look at some mushrooms?
I don't see any questions in the chat, Bill. Does
Okay.
anyone have any questions so far?
Can I speak up?
Okay, we'll just move on then. Right. So here is a parasitic fungus. It's a honey mushroom, and it's sprouting from the base of this birch tree. It's a virulent root rot fungus, and it's considered a serious pathogen, perhaps the most serious of all fungal pathogens as far as forestry is concerned. Now, honey mushrooms, it used to be thought there was one species of honey mushroom, but we know, and we thought, and it was said, well, some of them are brown, some of them are yellow, so forth. Now we know there are lots of different species, maybe 12 or 15 in North America. So here's another, the brown form that would grow around here. And it's growing here at the base of an oak tree, and you can see it looks different than the yellow one. So this is probably Armillaria gallica. The names change very often in mycology, so it's, you know, you can often see a half dozen names for the same exact species. But here it is. It's rotting the root of this oak tree. See, it's on the ground there. So the other thing about, some of you want to eat honey mushrooms, they're good to eat if they're well cooked. If they're not well cooked, they're going to send you to the hospital. Guaranteed. Guaranteed. Okay. And it means you don't do a stir fry with them, but you put them in your spaghetti sauce and you cook them for 20 minutes or so. So what we've been looking at there, when we talk about mushrooms, I'll just back up here a second. You remember from the first talk, these are fruiting bodies. These are just the fruit, like an apple on an apple tree. The actual fungus itself is the mycelium. And here the mycelium is bound itself together in these shoestrings. It's called shoestring rod or shoestring rhizomes. And after this tree has been killed, they feed on that tree. So it was a parasite, but now they're feeding on that tree, and they can take that energy and they walk across the forest floor until they find another tree to attack, or they might find your garage to attack, or they might find a dead log in the ground, or they might find some plants in your garden to attack. Okay, some of them are very large, and there's often these competitions. What's the largest organism in the world? And honey mushrooms are often given one of those nods because they form huge growths across whole sides of mountains. So when the tree falls, it becomes colonized by a complex ecological community, and you can see here a rotting tree. There's the standing, it's rotting. So lots going on inside that tree. There's two kinds of rot now. So now we're moved from the parasitic to the saprophytic function here. There's white rot, and you can see this is a birch tree, and you can see this is a fungus on it. You don't know it, but it's called Trichaptum biformis. This will be called Polypus parjaminus. It's a purple hymeneum that has tubes, but they break up to form teeth. Therefore, it's called biformis, two forms. But what they do is they digest everything. They digest cellulose, hemicellulose, and lignin. So what's left is white wood there. And they continue doing that until there's not much left. So here's the typical stringy pattern of decay from white rot fungus. You know, you've probably seen this on some of your walks, and that's, you see that, that's a white rot fungus doing that. Here's maple, which I found last year near the Phoenicia Reservoir. And it was attacked by a fungus called Burkhandira adusta. And it was all over the surface of the tree. But you see, a wind came and just snapped that tree in two. Just was so weak, it had no support whatsoever. It snapped it in two. Here is the fungus itself over here, and it was just covering this tree as if it were bark. You can see how white that is. That's typical white rot fungus. You'll probably find woodpeckers, when you find woodpeckers working in the forest, they like to work on white rot trees that are infected by white rot fungus because the wood is softer, there's more insects that live in it, and there's easier on their headaches, I guess. So in terms of numbers, there are most of the lignicolous fungi which digest wood, are white rot fungi. About 80% of them, maybe about 1,500 species in North America. So most of the fungi you see on trees are going to be white rot fungi. And most of them used to be called polypores. Here's one which is, those of you who are interested in eating, you'll want to look for this mushroom. I have collected it every month of the year at this latitude, around here in the Mid-Hudson area. It's called the oyster mushroom. There are a number of species of it, but it's like Pleurotus, the genus Ostriatus oyster, and related species. There are a number of them, but they're all pretty much the same. They're potent white rot fungi. They degrade the organic matter in the wood, the cellulose and the hemicellulose and the lignin. And they want more than that, so they make little nooses and they capture nematodes that are there, and they get an extra amount of nitrogen in their diet. And so that makes them have a sort of fishy smell. That's why they're called Ostriatus, like oysters. They are absolutely delectable. Those of you who have never tried that, look for them and make sure you have a good guide, and you're not going to get something that's going to make you sick, but that. So because this mushroom can digest lignin, we use this to digest other things that like DDT and PCBs and oil spills and sludge. So this mushroom is grown on sludge fields. Usually what happens, they put a lot of sawdust or wood chips in it, and then they inoculate that. It often takes about 4 times as much of the wood chips as it does what you're degrading, want to degrade to get this reaction going. But one of the good things about it is because they break down they do their digesting outside their body, they break down all that toxic matter, and what they bring into their own bodies is edible. It's not toxic any longer. So that's why we want to remember that the fungi do their digesting outside of their bodies. And many people in South America now, where they're doing this, are using the mushrooms that they get not only to feed themselves, but as a sale crop, cash crop. Now, this used to be called Polyporus sulphureus, but the genus Polyporus, decision was made to only include white rot fungi in that genus. So this mushroom is now called Lycoperdon sulphureus. And sulfur shelf mushroom. A lot of mushrooms are called chicken, having chicken in the name. Sometimes it's called chicken mushroom or chicken of the woods. You get a lot of different names there. So this is called Lycoperdon sulphureus or Polyporus sulphureus or sulfur shelf. And this mushroom does not digest the lignin, so you get a lot of brown rot. And this is what the tree that it's feeding on looks like. It sometimes has this cubic shape, and that's called cube rot. And what happens here is the fungus doesn't use so much enzymes that break down the sugars, but they use hydrogen and magnesium peroxide to literally burn their way through the tree. So this is the combustion here. And they'll follow the grain of the tree or the pores in the tree, the porosities, and they'll make that cube rot. In time, that rot will break down and make the soil. So brown rot fungi are really good for building up humus in the soil. And I've already mentioned sequestration. They take the carbon which is stored here, and rather than releasing the CO2, they move it into the soil where it's there as humus, a sustainable release. So it's a nice long-term solution for this. This happens to be a pine tree. I look at it, a pine tree. Here's another pine tree here, and this is a spruce. And what we have here is Lenzites gloriophyllum cepiaria. Used to be called Lenzites, but when we separated brown and white rot, had its own genus here. Only about 20% of wood rotting saprophytic fungi in North America are brown rotters. It's only about 250 species, but they're really important in the not only in the temperate forest, but more so in the boreal forests of Canada and through the taiga and in Northern Europe and Asia. These are the ones that will create a lot of soil there. Now you think about this. I'm trying to find there we go. Just think about this. Where we're living here, and I go north to the Laurentian plateau, the glaciers periodically would scour that area. So you get down to just bare rock. It's called the Canadian Shield. And all of that dirt that was there, all that the gravel and the rocks. Well, the gravel's around here. All these gravel pits around here. That's where that gravel came from. And that's where Long Island came from. And that's where Cape Cod came from. All the way down to, what's that called, at the southern border of Pennsylvania? I can't remember it now, but they dragged all the soil down there. Actually, if you go over to the Shokan Reservoir in the summertime when the water's out, you'll find lots of rocks there just scoured by glaciers. Or you go to the top of the Catskill, the peaks of the Catskills, you'll find rocks there scoured by glaciers. Right here where we're sitting now, there was a mile-high layer of ice here that just pushed all the dirt south. Well, how do you get dirt back there? Turns out that these fungi put it back, and they put it back in exactly the place where it's needed. So that's a real interesting look at how it is that the systems work and how they self-correct in a way. Here's a fungus which has intermediate rot characteristics. This is Lycoperdon Piriformi, the pear-shaped Lycoperdon. Lycoperdon means wolf's fart. So this is a pear-shaped wolf's fart here. And they depend on something to get in the wood previously and break it down a little bit. And there are a succession of fungi which grow on wood. So once that process gets started, this mushroom comes in then. It is a very good edible. It grows in sheets like this. And if you cut them open from top to bottom, if it's creamy white like cottage cheese inside, it's good to eat. And I know one of you said you ate the Calvacia gigantea, that big puffball, which tastes like nothing, like cardboard. This one has a good taste to it. So you'll if you like mushrooms, you'll like this one a lot. They're small, but you can make nice things with like mushroom loaves. Here it is, growing on this log here. It's just growing and growing and growing. It'll stay there for years. The log in the background is black, and that's brown rot fungi working on that. So that kind of decomposition can go on for a long time. Those of you who like to work with wood might look for logs that have this kind of patterning in it. It's called spalting. And what this is caused by is as the fungi are on the outside and work their way inside and start to rot, they start competing one with another for hegemony in there, and they build up these hyphal walls. This is my area. This is my area. Well, this is mine. Well, this is mine. And so that makes this wonderful patterning there. And if you do any carving or if you do bowl turning, this is beautiful wood to find. So here you find on the outside some turkey tail mushrooms and some parchment hard at work on breaking down this log. And they just continue. And after a while, here we still have that, that parchment here, Sterium complicatum. And as it continues to rot, porosities will develop. There'll be holes, and other life forms will take up residence. And those of you walking around, these are good places to look for things like newts and snails and all sorts of critters like that. Snakes love it in there. Squirrels, mice, flying squirrels love it for nests. So brown rot is particularly well suited for conserving moisture, and it's one of the reasons that we don't like to, in forests and climax forest, you don't want to remove all the dead trees because, among other things, they store a lot of moisture. And when you have a fire like occurred over at on the mountains, what was it now, maybe 8 or 10 years ago, that big fire that occurred there. If you have a lot of down wood on the land, it often can act as a fire break and stop that. Between here and there, however, it serves useful functions in the ecosystem because it's places where you have a lot of algae going on. There might be slugs in there, snails, insects, litter, eggs, seeds. Here's we have a slime mold. Here we have a little basidiomycete growing there. So a huge complex starts forming their own little ecosystem in here, their own little community here. So you have the ecosystem is comprised of a lot of these different kind of habitats and different communities. And brown rot is, we talked about it for sequestering carbon. Here's really good to support some of the complexity of the ecosystem. It's said of saprophytic fungi that they are nature's primary recycler. And they actually existed before, well, I think they came out of the ocean at the same time as plants. It's the current thinking about that, but they're the basic recyclers. So here we have one growing on wood. Fungi growing on wood are referred to as lignicolous fungi. And this is Vulvariella Bombasina. It is a choice edible. It's related to those Chinese straw mushrooms, but this one grows to enormous sizes. This could be 8 inches across. Grows just like an amanita, but and actually it's a Proteaceae, so it's related to amanita in that way. But it has free gills. It'll have pink spores growing on wood. I've only found it 4 or 5 times in 60 years of professional collecting of mushrooms. But it's wonderful to behold. It's just a wonderful, wonderful thing to see and and very tasty. Here's a mushroom which has become quite common in the area. I first collected this probably in 1970. It was just coming into the area. It's not a native. It's been introduced on some sort of woody debris. It was found in New York City environment first, then it's moved all over the place. And now you find just about every place where, well, maybe every place, about half the places where line crews come by and they chip up wood and leave the wood chips there, you'll find this growing there. It's called Stropharia Rugoso annulata, and it is a superb edible. Here it is growing on wood chips in my garden. My garden was an old railroad track, and it now has soil which is about 18 inches deep, just deep loam. And I've grown that there by putting lots of wood chips there and introducing these mushrooms that break down the wood chips to make that wonderful humus that's there. And these are very good mushrooms to eat. Other mushrooms, the saprophytic mushrooms are terrestrial, and this one happens to be toxic. Terrestrial means they don't grow on wood, but they grow in grass. So this is Agaricus placomices. It's related to the common store-bought white mushroom, but it's different. And it grows in grasslands, a place like this. I don't know if that was growing here or not, but here you see a fairy ring. And the fairy ring is a place where recently, last year, mushrooms fruited. And in growing and fruiting, they took the carbon, the dead grass that was in there, and they recycled it and they put the nutrients back in the soil. So that's how you get this kind of fairy ring. Originally, that fungus was right here, and it just grew out year by year by year by year by year by year, making a longer and longer circle. From satellite imagery, you can see huge circles like this up where there's Stonehenge and places where there are big prairies, where mushrooms have been growing for thousands of years. So knowing this, if there's a mushroom here that you collected in this field and it was edible, and next year it's going to be out here. Just keep growing there. And they grow up just very quickly. As soon as they get enough water in them, they just balloon up very quickly. Here's a mushroom which loves dung. This is Caprinus cinereus. Here's puffballs, and they're great recyclers. There's about 3 in the area that look like this. This is Craniformis. It looks like a skull shape. There's Vasiformis. It looks like a vase shape. And there's Gigantea, which looks like a large soccer ball. They can be distinguished not only by the shape, but they have different color spores inside. So long as the spores are white and no trace of brown or purple or yellow or green, they're okay. Okay to eat. They're not that delicious, they're okay. And then finally, we have Morels. And Morels use both a saprophytic and mycorrhizal feeding strategies. So that brings us, we can start talking about Mycorrhizae. This is Morchella esculenta, the common yellow mushroom. But before that, maybe Lauren wants something to say about the program we have scheduled on Morels.
Yes, so coming up in April, as you can see maybe at the bottom of the slide there, on April 22nd, we're going to, Bill's graciously going to host another webinar all about morels. So right now you're learning about the ecological importance of them, but he's going to talk more in depth about these awesome mushrooms.
Right, and where you can get them and how to look for them and lots of stuff like that. Yeah, I think I mentioned, I think last year that New York Mycological Association that drew 400 people. They were all going to go out to Central Park. Not all of them. Okay, so now we're on to mycorrhizae. It's a hugely important area. There's 2 types of mycorrhizae which we'll talk about. Mushroomers are almost always interested in ectomycorrhizal fungi. They're in temperate and boreal forests. They include mushrooms that grow on top of the ground, basidiomycetes, truffles grow under the ground. Here's something important. There are 5,000 species of ectomycorrhizal fungi. I mean, give or take 1,000. These numbers keep changing all the time, but a lot of fungi. They service 2,000 plants. That's quite different from the endomycorrhizal. Endomycorrhizal fungi, there are 300,000 species of plants that are serviced by only 120 fungi or thereabouts. So they're very different in the way they associate and the way they act. So here we're talking about glomales, almost all ascomycetes. There are several other kinds of mycorrhizae which are discussed. By and large, they are variations of endomycorrhizal fungi. Those of you who are interested in this can go, and this is a wonderful site here, mycorrhizas.info. It's from Australia, but it's a wonderful introduction to mycorrhizae. Okay, so let's start looking at mycorrhizae. Here is a root of a tree, or shrub, plant, and it's comprised of the harder inside, the stele, and then the cortex, and then the epidermis, okay? And from the epidermis, some of these cells become root hairs, and they don't go very far into the soil, maybe an inch or 2. What happens with the root hairs is that hyphae from fungi will come and they will invade the root, usually through the root hairs. They'll invade it. Now, the endomycorrhizal fungi, they're often called VAM or VA mycorrhizal. V for vesicle, vesicle is like a sac. A for arbusticle, like a little tree, like an arbor, like a little tree. So the endomycorrhizal fungi, actually, their hyphae penetrates inside of the cells of the root. And here you see them here inside the cells of the root. and where they are going to exchange nutrient material with that tree or shrub or grass. Ectomycorrhizae are somewhat different. You have the same thing, the root and tiny root hairs, but here the ectomycorrhizal fungi form what's called a mantle, a sort of corky layer all around the outside of the root, and it's often called a heart-teg net, where they interface with the interstitial space between the cells. Okay, so there's two very different ways. You can see them right here. Here's the mantle and the heart-teg net there. Now, one of the ways to think about this is that this is controlled parasitism, because in order to get in, the fungus has to eat part of the walls of the cell to get in. And in order to get the food back, the tree has got to destroy the fungus and eat the fungus. So I remember in the 1960s and 70s, and Goldman will remember this, there was this American Zen master, Alan Watts, and he used to say that that nature is a mutual eating society where we just eat one another all the time. And that's what we now would call controlled parasitism. So here would be an endomycorrhizae. Okay, here's where the fungus has eaten the cell wall and entered, and this arbusicle, its fate is limited because it's going to be dissolved by the plant in order to get the nutrients. In the meantime, once inside, the fungus is sucking all the material out of the plant in order to feed the fungus. And we'll look at that in just a second. Now, sometimes something very interesting happens, and the fungus adapts to the internal environment and gets cut off from the outside world. At that point, it becomes what we call an endophyte. Okay? Relatively new term in mycology and biology, but it means inside of a plant. Can infect a fungus or a bacteria. At least part of its life is spent in there. Usually it's passive, it doesn't cause any disease at all. It often enhances plant growth that protects it from pathogens. May also be a latent pathogen or saprotroph. After the plant becomes stressed or gets old, it then helps kill the plant. It does alter the chemical production within the plants, and that offers some protection from herbivores, and there are useful agricultural and medicinal uses that flow from this. And it can be transmitted vertically or horizontally. Now, I don't know if you're familiar with these terms. Vertical transmission of genetic material means that the genes you have come from your mom or your pop. So, and so you've got your grandparents, your parents, and you, and then you're going to give that on to your children and they to their grandchildren. That's vertical transmission of genetic material. Horizontal transmission occurs by what's called usually plasmid exchange. Inside the cell mechanism, little plasmids break off. Often when the chromosome zips and unzips, some RNA will break off. Actually, that's, if you think about that, the current vaccine we're all waiting for is an RNA transmitter. So it's part of that process. So you get this horizontal transmission. And the way you get that, actually the way the coronavirus occurred was that it was in bats, and bats were put in close conjunction apparently with pangolin, and it transferred from one species to another species, and then it transferred to humans. HIV transferred from SIV, simian immune deficient virus, into humans. And so that's a horizontal transmission of genetic material. So that occurs inside of the living cell of a plant. So more typical in Ascomycetes and grasses. Here, what you can do, there was some talk about using mushrooms for medicinal purposes. By and large, the critical literature does not support that. I know the folk literature does, and a lot of homeopathic and people do, but by and large, the critical literature does not say that if you eat a mushroom, it's going to cure an illness. You can do that, but it takes some working to do it. For example, if you have breast cancer, you can take this hyphae right here, which is stained by cotton blue. You can pull it out, and you can use all sorts of techniques to bulk it up and to purify it, and then to spread it on agar plates to watch it grow, and then use that to test different chemical properties. See, for example, if it kills cancer in culture. And when it does that, then you can play with that some more and develop a medicine like Plaxitaxel, which came from the yew trees, but there weren't enough yew trees, so now they're making it from bulking up these little threads that occur in every leaf of every tree, we're told. It's probably hyperbole, but it just goes to show you they're ubiquitous, they're all over the place, and those are the techniques that are useful in curing diseases and in medicine today. So think about back to VAM and Ectomycorrhizae, the vesicular or vascular mycorrhizae, which grow inside of the cells. They don't form mushrooms. They form large spores, but there's no fruiting body. With the Ectomycorrhizae, they form mushrooms, and the spores are tiny spores which are on the hymenium of the mushroom. So let's have a look at some of those. Oh, before we want to do this, I guess. This is what some of the mycorrhizae look like. These are what are called the morphotypes. If you dig in the soil, that's what they look like. They vary, they're difficult to isolate and identify. And DNA evidence indicates that several fungi share the same mycorrhizae. And again, that's another important place where you're going to get plasmid exchange and transposons. So these things are ubiquitous. So by the numbers, there are relatively few mycorrhizal fungi that can be identified by field collections. Usually I teach mushroom identification in courses, and it's very difficult. Those are difficult for people to learn, for me to learn, for anyone to learn. You know, there's maybe 3,000 to 5,000 species of fungi around here. You know it's a mushroom, but what species is it? Well, I don't know. You know, it's hard to tell, particularly by field collections. Now, if you bring it home, you have if you have some literature sources and you have microscopes and chemicals, then you can start to identify it, but to recognize them, that's kind of difficult. In the same way, a few of these can be recognized or identified by morphotype, by what the shape of them, what they look like. However, now that with DNA techniques, you can screen the soil or the water or fecal matter and find all sorts of previously unknown genotypes. I mean, they're finding new kingdoms now that no one ever suspected there were these kingdoms. And they're not looking for any fruiting structures. They're just going in and looking at DNA, okay? And so now with science, you know, we've changed the method by which we define things, and you really changed the whole meaning of a lot of what we thought we knew at one time. I mean, it's not that we thought we knew it, we still know it, but there are different levels here of what's going on. It's all still systemic, it's all part of the ecosystem, just getting more and more refined in how we're looking at it. This particular slide here was one of the original studies about what we call the wood wide web, and it was this particular garden vegetable and this particular endomycorrhizal fungi, which was studied. So here you see the same thing. The hyphae are tiny, tiny, tiny. Okay.
Bill, we are at 8 o'clock. So if you just want to hit a few more points, we can open it up for questions.
Okay. We still have a lot to go here, don't we? Okay. So the thing about the mycorrhiza is that they extend the rhizosphere of the root itself. And here you see a seedling, and what you've got here is other seedlings which are planted. This one apparently is nursing the others, from which the term nursery came. And Now we have mycorrhizal ecology. Maybe it's a good place here to see if someone has a question before we go on.
Sure, sure. Are there any questions before we hit just a few more slides?
I'd like to ask a question.
Sure.
Can you talk about like the etiquette of collecting mushrooms in the field? Like, are we allowed to do it? Like how does it go about?
Yeah, there's both legal implications and others. In state forests, you're not supposed to collect anything. Since mushrooms are the fruit, it probably doesn't hurt. I had to check this out with DEC several times, but it probably, they as long as you're not causing a nuisance, they're generally not going to do anything. It's like picking blackberries, but the etiquette is you don't want to pick them all. You want to leave some for spores. Lauren, I have a question. Are we going to go on to 8:30?
Well, I think that we should probably wrap this up pretty soon here. As I know people maybe have some other things to do this evening. So if you just want to hit a few more quick points and we can open it up.
Okay, I guess we hadn't communicated. I thought we were until 8:30. That's where my speed was going along here at this. I was enriching all of these. Well, can we make a compromise and those who want to leave can leave and I'll just continue talking and they can look at it later on the recorded version? How do you want to do that?
Yes, this is being recorded, so we can continue on.
Okay, good. All right. So the basic features of mycorrhizae is that the tree shares with the fungus carbons, sugars, and vitamins, and in return, the fungus brings largely phosphorus, water. There's a heat and drought buffer, and they protect against pathogens. And it's about 95% of plants or half of all forest biomass is thought to be wrapped up in this process. These are often called, fungi are often called mycoheterotrophs, that this is such a symbiotic relationship that they cannot be separated. So here, for example, is one fungus, is Swillus pictus. It grows with white pine. White pine will not grow without it. And so those two make a very clear mycorrhizal symbiotic relationship. Here's a generalist. This is Amanita multisquamosa, part of the Pantherina family of Amanita. And it can service oaks, poplar, pines, lots of different species. This is the Bokitis edulis, the very popular king mushroom, the porcini. It's a prized edible and it grows with mycorrhizal trees. The expanded view might be something like this. You have here radiant energy from the sun. We see Roy G. Biv, but there's a spectrum on either side of that. And photosynthesis captures that energy. And what it does is takes carbon dioxide from the atmosphere, splits it up. It takes carbon dioxide, splits it up and gives back oxygen, and it stores the carbon in the organic matter. So organic chemistry is a study of the carbon molecule. So you have the carbon here, and the carbon in this tree here will go down and will feed what we call hyphal mat communities. It'll feed the mycorrhizal fungi, which feed little saplings and little trees and whatnot. So the fungi are here, they're releasing enzymes, exudates come. What you got here is a lot of a decay that goes on. There's bacterial decay, organic recycling. There's sometimes some fungi will fix atmospheric nitrogen. Largely that's bacteria that does that on legumes, but fungi will do it as well. So what you got here then is this goes on and will start to feed animal communities. Oxalic acid caused by the fungi will release phosphorus from rocks. And so you get the phosphorus coming back up here. You get water coming back up here. You get heat and drought pathogens. And larger, this whole system stabilizes the water tables. It controls erosion. It detoxifies soils. It inhibits non-mycorrhizal plant competition and increases nitrogen availability. And not to forget up here that the trees are raking the clouds and fog, and you got nitrogen falling down here, nitrogen and water. So from any point of view, you can call this the part of the nitrogen cycle, the carbon dioxide cycle, the carbon cycle, the oxygen cycle, the plant cycle, or the fungus cycle. Lately, there's been a lot of emphasis on fungi, and there's a lot of popular literature about that. So here are some of the fungi which are partners in this mycorrhizal community. This is a russula, and the one in the back, the green russula, and this is the Bokitis frostii, really beautiful fungi. These are chanterelles, great edibles, and most of them are mycorrhizal. This is Cantharellus sabarius. It's probably the best of them. This is a common truffle of the area, Elaphomyces granulatus. Grows under hemlock, and you can see it's about the size of a penny. Now, here, some of you have seen these plants like this, Indian pipes. This is Monotropa uniflora, one flower, turning over once, and it doesn't have any chlorophyll in it. So how does it live? Well, there's a particular group of fungi, not all of them, but a particular group of fungi which it hitches a ride to, and that fungus is making its living off a tree, and it's bringing it to a mushroom, and this one just happens to be midstream capturing that energy. So that's how those achlorophyllous plants survive. There's a wonderful edible, Bokitis bicolor. I find that under birch and oaks. It's a wonderful. As long as it doesn't stain blue. Some of the ones that stain blue are not good. This one in particular, this one is Bokitis subvolutipes. Subvolutipes means it has this less than hairs here, velvet, less than velvet here. And I know this particular fungus was growing in a cluster in front of the Gifford House at Cary Arboretum. And someone came in and they broke off a corner of one of them and ate it. He spent 3 days sick, I think a day in the hospital and 2 days at home recovering, just from a thumbnail part of that mushroom. There's a lot of talk lately in popular mushroom talk that, oh, you can eat this mushroom. I never have, I never will. I work with poison control for some 30 years, and there's a number of cases of people who have eaten this who end up sick. So I will just not eat it at all, and I don't recommend you do it either. So it's got red tube mouths here, and it stains blue instantly. A few mycorrhizal fungi are deadly. This is Amanita bisporigera or verosa, and it's also often called the death cap, although that's probably a corruption of death cup, because it has this underground cup. So it's white all over, has a ring rather at the top, the annulus here. The gills are free from the stem, and as this structure grows on below ground, and it is deadly. It'll kill you. It takes about 5 to 7 days to do it. It essentially stops your liver and kidneys from functioning, and then does a number of other things too, red blood cells, and then the whole cascade that falls from that. So here, this is the underground cup, the death cup. To identify any amanita, you have to go underground and start the identification there. If you just cut a mushroom off here and don't, you very easily can end up with something you don't want. So just a little bit more fungi growing on fungi and fungi growing on plants. These are pathogenic. This is a lobster mushroom. It's one fungus growing on another. Although it's pathogenic, it is wonderful. It is one of the best mushrooms you're going to taste. It smells the smell like lobster, so it tastes like lobster. It has that lobster color to it as well. What it is this mold which grows on a species of Lactarius. So it's Hypomyces, hypo on a mushroom, lactifluorum of it'll lactate, it'll give you milk. Choice mushroom. The foray in Maine once we had, several, we were lucky to find probably 50 or 60 pounds of this, and we had lobster right from the ocean. So it was lobster mushrooms and lobster, and what a meal. Wonderful. When you go to a foray, the mushrooms are brought in and they're spread out on tables, and almost always you're going to get this Sporidia grandis growing on the mushrooms, called a troll fungus, right? Has a number of names. Syzygites megacalarpus might be a name that's still common now, but it's a mushroom growing on another mushroom. Here is one mushroom growing on another mushroom. It's a cordyceps, which is growing on a truffle, on a Laphomyces granulatus. And so you just see this ascomycete sticking up, and the spores are little pits on the surface right there. These are often collected for medicinal purposes. This is a pathogen on a plant. This is Phytophthora Infestans, the fungus responsible for late blight of tomato or Irish potato famine. Used to be that we only had one, either the plus or minus strain north of Mexico, where the fungus and potatoes and tomatoes originated. But with the advent of the big box stores, a lot of things were brought up from the tropics, and now we have this. They have it's now and when it was only positive or negative, it never could form a resistant spore. Now they can form resistant spores and live in the ground. So it's a perennial problem we have now. They take place, grow quite quickly. A single cell from one of these lesions can float through the air, or if it lands, it can swim. It has both flagella that enable it to swim, and it can float through the air. When it lands on the leaf, it can either digest the leaf or go in through an osteo, a little pore there. So once it gets in the plant, it takes a day or two, maybe 3 days to mature, and then it starts to release 300,000 spores a day. So within a few days, you can have the complete destruction of your entire potato crop or your tomato crop. And those of you who garden around here might remember about 10 years ago, there was the first really episode of Phytophthora Infestans just killed all the tomatoes around. The spores will go 50 miles, so if anyone in your neighborhood gets their plants from Target or Walmart or wherever they're selling them, chances are you're going to be, even if you're growing organic stuff, it's going to be infected. By the time it gets into the stem like this, the plant's gone. Endothia parasitica, the chestnut blight can get into small saplings and get into chestnut, and it causes these chancres, these blisters to erupt, and the tree dies. So it never makes a really healthy seedling, so the crop dies. The roots live forever, but the tree itself dies. And that brings us to another talk we're going to have here on destroying the American chestnut. Lauren, you know these are people from Mohawk who are doing this, is that right?
Yeah, there's some research that's been done by graduate students at SUNY ESF, and next week on Tuesday, January 26th, they'll be giving a presentation on restoring the American chestnut and some of the work that they've been doing here on Mohawk Preserve. So pretty excited about that one. There are still spaces available, so if you are interested, please do sign up for that one.
Oh, great, yeah. This is another pathogen. It's an interesting kind. It's Gymnosporangium Juniper Eye. It grows on red cedar, the juniper, and then it looks like an apple growing there. And then the first warm rain in May, these sprout long orange fingers, and they spore, and then they sporulate, and spores go and infect apple trees. And on the leaves of apples, it looks like little cedar trees, so it's apple cedar rust. And it's a heteroecious fungus, requires 2 hosts. And you remember from the first program, there are many like that. So if you want to protect your apple crop, very often people cut all of the cedars down around them. This is an interesting one. This is Ustelago matus, or corn smut. The Mexicans call it huitlacoche. Here it is growing on an original type of corn. The corn is a double husk corn, and it seems to prefer that. has this black maturing spore mass here, but inside it's white and creamy. I've never eaten it, although I grew this in my garden, good out in my garden. But this I've never eaten it. But you can get it in Mexican restaurants and in canned goods stores in Mexico. Here's a Cordyceps variabilis. It fruits another Cordyceps, but this fruits from the head and only head of an insect pupa. And there are a number of insects that this occurs on, and it grows only from the head. And some people call these zombie fungus. Some of them actually, the ants will crawl the highest part of a tree, and the fungus will grow out of there, so it spreads its spores very far. And this is Schizophilum commune. It's been known to affect humans and form mushrooms on, I've seen photographs of mushrooms on fingernails, on the hard palate of inside the mouth. I've seen this mushroom growing as a mushroom on lung tissue inside the lungs. This is the underside of it, and this is the top of it here. It's often used for genetic studies because this one has about 28,000 sexes, if you remember from the first program. If not, you maybe want to go back and have a look at this. Okay, so what this brings me to the end of the talk here. We've talked about ecological functions of functions. Oh my goodness, did I make a mistake there? Functions of fungi, the parasitic, saprophytic, mycorrhizal, pathogenic. We talked about endophytes, symbiotic relationships. Fungi are often now called mycoheterotrophs. The fungus and the tree it's growing with are one organism. There's another environment in Maine, and that brings me to the last slide here. This was the second of the three or four talks. There are a couple more we're going to talk about. Instead of morels, we're going to talk about, instead of spring mush, we'll talk about morels, but common edibles and their toxic lookalikes is another, and I'm working on one on the Wood Wide Web. So there's other possibilities if there's a desire for that in time, space, and all of that sort of things. But here in the midst of COVID, we've had two of these talks here. So I thank you very much. I'll stay around here for any kinds of questions anybody wants to have. Let's take it from there. What kinds of questions do you have? Anybody there?
Sure. I don't see any questions in the chat, but if anyone does have a question, please do chime in, and you can put it in the chat, and we can see if we can answer any of your questions. And again, thank you, Bill, for this great talk.
Well, you're more than welcome. This has been fun. Yeah, I'm trying to get rid of this screen here. There we go. I guess get rid of that screen. So anybody there still around?
There's a few people still around. You're getting a lot of thank yous in the chat.
Oh, good. Yeah, I'm trying to get pictures back up here. Where's Bill Stickler and Nancy and Amy? And
Sure. Yeah, you can go up to the top of the screen there, or actually down at the bottom where it says share screen. You can say that you can stop sharing your screen.
Oh, I can stop sharing that. Yeah.
Yep.
Oh, there we go. Right. Thank you.
There we are.
Okay.
Yep.
So I see you. You can see me.
Thank you, Bill. This was amazing. So.
Okay. Okay, Richard, give me a call at home. Wes, same thing.
Yeah. Well, thanks everybody for
question.
coming. I don't think anyone has any questions. We're getting a lot of waves. Goodbye.
Okay.
Thanks, Bill. It was great to see you.
Yeah, same here. Right. Really good to see you. Right.
Take care.
I've got a question that's taken me forever to type it.
Oh, okay. Just go ahead and talk it.
All right.
I
so-
have the same problem, incidentally. I don't type so well, but yeah, what is it?
Yeah, well, in the beginning, or near the beginning, you talked about the idea of a possibility of a tree living forever. Now, doesn't that stem from after the tree is falling and being eaten? Doesn't it stem from the possibility of that same part of that tree growing out of the stump and being able to take root once again?
Oh, that could be. Yeah. That happens a lot. You see that a lot where the tree is cut and the cells there they're growing again. Yeah, but those cells would come from the outer layer of the tree, That's the only part of the tree that's living. The inside of the tree, the heartwood, is dead. And it's protected from decay by the living sapwood and the layers on the outside. So that they act sort of as a
So is that the Cambrian?
the Cambrian layer, yeah. The phloem and the xylem, and they have a lot of just like we have a lot of vital parts of our body that function as an immune system. They do too, and they stop fungus there or the bacteria. So if you cut it with a lawnmower, then you get rot or something, decay in it. Yeah, but what you read is that it's potentially immortal, and some trees, are 5,000 years old, so they do live a long time. Yeah.
And one other thought, if I may, is that all right?
Sure, yeah.
The second branch of mushroom, not the endos, the
Ectomycorrhizae?
yeah. Aren't they being taught to eat? When you talk about mycoremediation, aren't they being taught to eat oil spills and such, and even haven't they been found at Chernobyl? Is that true? Do you know anything about that?
Yeah, and Chernobyl is quite interesting. Fungi pick up heavy metals very easily, so they pick up the strontium quite easily. And actually, it wasn't just around Chernobyl, but you find strontium appearing in mushrooms up in Norway, for example. But they do pick it up easily, and they appear to be able to tolerate that. Some better than others, they tolerate it, and so they actually can adapt to that sort of environment. Yes, there are fungi which are mycorrhizal with blueberries, which do the same thing for lead. They pick up lead. Actually, one of the problems about morels is they pick up lead and arsenic very easily. So they'll pick them up quite easily. So yes.
So that's transferred to the human?
If you eat morels that are full of lead, yes, that you'll have lead poisoning from them. Yes.
So how do you avoid that?
You don't pick the ones that have lead on them.
How do you know?
Tune in when we talk about morels, and we'll talk about it. Or actually, you could Google my name and morels, and you'll find several articles about that.
Right, and that first part that I was saying about the being taught to eat some unmentionables, I've seen pictures of them. They were taught to eat cigarette butts.
Yeah. Oh, yeah. Sure. Yeah. Mushrooms will degrade anything organic. Yeah. And cigarette butts. Oh, the one you're talking about. Yes. They. Right. I'm remembering that.
Sheldrake's book.
Merlin Sheldrake's
right, and Sheldrake talks about that. They are. What happens is the concept in biology is called genetic drift. If you imagine that bell-shaped curve that we talked about, nature tends to distribute itself on a bell-shaped curve. So most of the mushrooms are average when it comes to being able to tolerate, let's say, to digest this kind of cellulose or whatever it is that the cigarette butts are made out of. Some are better. So if you feed your population of mushrooms to this cigarette butts to them, some will do better than others. And then if you take those and cultivate those, again you'll get a bell-shaped curve. But now the whole population will be somewhat stronger than the original population. If you keep that up, then after a few generations, you can get a fungus that can do what you want. Maybe you've seen or maybe you've heard of experiments with fox in Russia. The fox were raised there for pelts, and because they're caged, they're very, very angry. But every now and then you find one that was more docile. And what they did was they took the more docile of the fox pups and they bred those together. And in 4 years, they turned the fox into dogs. They changed their color, they changed their behavior, they changed the chemical composition within their body. That's what did all that. They changed the length of their snout, and they became little tail wagging puppies. And that was only in 4 generations. And that should be easily Googled too. So that's just understanding how nature works and taking those laws and putting them to use to do that. It's not that the fungus has a knowledge, that it's just that you're taking advantage of some natural phenomena.
Well, speaking of which,
Let's see it, Sandy. If anyone else has a question, and if not, we'll come back to you. But I like talking better than typing. Yeah. Anyone else there? Laurie still around? Who else is still around?
Yeah, we've got just a handful of people, but we should wrap this up. So
We certainly should. Yeah.
yes. Well, thank you everyone for sticking around this extra long time here. And I hope that you come and join us for other webinars in the future. Like I said, there's another one next week on the American chestnut, and Bill's going to be kind enough to do another one in April on Morels. So I hope to see you all then.
Okay. Right. Bye-bye.
Bill.